Hi, can I please know if another class is there at S7 or something? Or can I take 1 hour?
Sir, I can't get you.
Is there another class at 7 o'clock?
Yes, sir.
Okay. Okay, then I'll start then. Okay, good evening. Today we're going to go to the next topic in your exam mastery live class sessions. That is about cell transport and apoptosis. Okay. So we'll be basically going through these basic important questions today, where we'll be talking about transport mechanisms, a bit about your factors affecting all of that, and a small note on your apoptosis. Okay, so cell transport is a very important question which keeps getting repeated. Okay, and another thing is when you talk about cell transport, it can come even as an essay question where you'll have to describe the entire transport mechanisms. It can separately come as a short note, for example, diffusion, the difference between facilitated and your simple diffusion also keeps coming. Then again, vesicular transport mechanisms like exocytosis, endocytosis, all of that again comes as a question. Phagocytosis by itself can come as a short note. And based on the number of marks that is given, it can even come as a long question as it's given over here. Okay, so based on that, you'll have to decide how you want to answer the question, how many points you have to write. And if it is a longer question, I'd prefer if you draw the diagram and explain the various steps that are involved over there. Okay, and beyond that, we'll discuss a few of these short questions that are given over here. Okay, so to start off, just take a look at the first question and enumerate various transport mechanisms across cell membrane. So if that is a thing, then you'll have to understand the entire outline of your transport mechanisms. Okay, so that is the basic thing that you have to do. And we're going to start off with understanding this quickly. So when you talk about transport mechanisms, we have basically two types of transport mechanism. We have a simple and an active one. Okay, so this first one is what we call passive. Another one which is called active. Okay, two types of transport. A very basic kind of transport, which is passive transport, and an active transport. In passive transport, the whole thing is that you don't actually require ATP. All of the movement is down the concentration gradient somehow. In active transport, you have to go against the concentration gradient, and hence it requires some sort of energy requirement, either directly or indirectly. Okay, passive transport can also be further classified into your simple diffusion, your facilitated diffusion, and we have non-ionic diffusion. Okay, so these are all different types of diffusion. We have simple diffusion, facilitated diffusion, non-ionic diffusion. Okay, so under passive, we basically have diffusion, which is simple, facilitated, non-ionic. On the other hand, active transport, we have a primary active transport and a secondary. So the main idea behind the difference between them is based on how they use the energy. So for transporting something against a concentration gradient, when there is direct use of ATP directly, okay, or some sort of energy, usually it's ATP, which is used directly, it becomes a primary transport. So we have one transporter which uses ATP and itself transports substances. That is what we call a primary active transport. On the other hand, a secondary active transporter is a little different. Secondary active transporters, what they do is they first go under the premise that something else utilizes energy and creates some sort of a gradient. Okay, most often in physiology, we see sodium potassium ATPase being used. The sodium potassium ATPase is being used, okay, to create some sort of a concentration gradient that favors now another transporter to allow movement of ions. Okay, so in a way, this is not directly using your ATP. Something else is using the ATP. But because something else used the ATP and created a concentration gradient, now this is able to move it along. Okay, so this is an example of a secondary active transport where indirectly against the concentration gradient, you're able to move things because of another protein which basically created that gradient for us. Okay, so when you write the short note or a long question like this, you first start off with giving a basic flowchart as to what are the basic differences between them and explain each under this. Okay, now we'll come to describing each of them because each of this can come as a separate short. Okay, and when you're writing the long question, this description of each of that, briefly describe them so that it becomes a long question based on the mark. Okay, so in simple, diffusion. What is diffusion? There is a membrane over here, semi-permeable membrane over here. There is a concentration of substances over here, and there is a slightly less concentration of substance on another side. Okay, Brownian movements are going to happen, and there is net diffusion based on the movement. There will be movement of substances in this direction, but because concentration is higher over here, the net movement is going to be in this direction. Okay, your net movement is going to be in that direction. That is what we call diffusion. Now, this diffusion can happen in a simple way. Okay, so for example, you take a membrane like this, and there is a lipid-soluble substance. Okay, so we have a membrane over here, and there is a lipid-soluble substance, and that lipid-soluble substance can move down the concentration gradient like that. Or on the other hand, there's a water-soluble substance, in which scenario, to go across a membrane, you'll require a protein, usually an ion channel. Okay, that ion channel is opened up. Again, it goes down the concentration gradient. So this is an example of a simple diffusion. Okay, in facilitated. So this is an example of what you call a pore. Don't confuse it with what I'm going to tell you now. In facilitated diffusion, instead of this, there is higher concentration gradient on one side, less concentration gradient on another side. Okay, this substance cannot pass through the membrane in any way. Okay, it does not probably have a proper set of ion channels also. So instead, it requires specialized carrier proteins, which is able to take this, undergo modifications. Okay, structural modifications, and flip and take it on the other side. So although this is going down the concentration gradient, it still requires a carrier protein. So that is the essential difference between simple and facilitated diffusion. In that, facilitated diffusion requires a carrier protein. Now, the thing about this is, as we were telling about concentration gradient, more the concentration gradient, more the rate of flow is going to be. Okay, when concentration gradient increases, the rate of flow increases. Okay, so if we were to plot, so again, as I told you earlier, one question that is often going to come is difference between simple and facilitated diffusion. Okay, so you have to give points as to what is simple diffusion, what is facilitated diffusion, then draw this diagram that I'm going to draw. Simple diagram, you have to plot the concentration of a substance versus the rate of flow. Okay, so as the concentration of the substance increases, concentration gradient increases. Here also concentration gradient increases. Okay, this is the rate of flow. Now, in simple diffusion, as the concentration gradient increases, the rate of flow keeps increasing. Okay, on the other hand, for facilitated diffusion, when the concentration gradient increases, rate of flow, yes, increases to a limit after which it cannot further, it's not that it goes down, it cannot further increase beyond this. It remains constant. The rate of flow cannot go beyond this point. Okay, so this is because of saturation kinetics, where this transport is limited by the number of carrier proteins which are available. Okay, so this is where the essential difference is between simple and a facilitated diffusion. Okay, non-ionic diffusion, on the other hand, is when a very common example that you see of non-ionic diffusion, okay, is H2CO3, for example, splitting into CO3 minus and H plus. Okay, so H plus HCO3 minus in this ionic form cannot pass through the membrane. Okay, although their concentration gradient is higher over here, they cannot pass through the membrane like this as such without any particular transporters. Okay, so instead, what is it gonna do? This is gonna go back to be H2CO3, and that is gonna split to form CO2 and water. Now, CO2, you know, is lipid soluble. This can easily go inside. Okay, pass through the lipid membrane, and over there, CO2 now combines with water to give back your H2CO3, which will now go and split into H plus HCO3 minus. So this ionic form, which was there, gets converted to a non-ionic form, which is able to pass through the membrane, and ultimately, indirectly, it gets inside. So this is an example of non-ionic diffusion. So the reason why I specifically mentioned this, emphasizing on this, is because what I notice is students are never able to properly write this point. So when you're writing a full long question, they tend to leave non-ionic diffusion part. Okay, and if it comes with a separate short note, again, students are not able to explain this well enough with a example. Okay, so we got simple diffusion, facilitated diffusion, non-ionic diffusion. Now we have active transport. Okay, so can you see here? Explain active transport with an example. Apart from that, you have a very important question that keeps coming up from this topic, which is not mentioned over here. That is your sodium potassium ATPase. It is often asked and repeated. Sodium potassium ATPase, you have to know. Okay, it can come by itself as an essay. Long question, short question. Okay, so this is very important in this topic to understand sodium potassium ATPase. So the gist of what you need to know is summarized over here. Okay, so you have to draw the diagram of sodium potassium ATPase. You have to mention how it has a alpha and a beta subunit. Okay, so in a membrane, it has a alpha and a beta subunit. Now, this alpha subunit is what you have to be primarily concerned with. In the alpha subunit, you can see over here, there is one area where ATPase activity is there. And because of ATPase activity, what will happen? ATP splits to give inorganic phosphate. That inorganic phosphate is now going to go and attach itself to a particular area, okay, where aspartate 376 of the amino acid chain of this alpha uh, subunit. So once it goes and attaches itself to it, it will result in configurational changes to your alpha subunit. Okay, so what happens is your sodium gets bound on the inner side. Okay, towards the cytosolic side, sodium is binding to it, and on the outside, extracellular side, your potassium is binding to it. And when the configurational change happens, sodium goes outside, potassium comes inside. At your level for your exam, this is more than enough to understand at this level. Okay, this process is very complicated, not at all like how I'm describing it. But this much is enough for you at your level. So again, you have to draw alpha and beta subunit and mention how sodium binds to the cytosolic end, extracellular end, potassium binds. Okay, how many sodium? 3 sodium. How many potassium? 2 potassium. Now there is the ATPase activity which is there, because of which inorganic phosphate is released, which is now going to bind to aspartate 376, resulting in a configurational change, resulting in this flipping. And when this flips, this sodium moves outside, potassium comes inside. Okay, so one short notice, describe the mechanism of sodium potassium ATPase. All right, if it's coming as a long question, you can mention the diagram of your sodium potassium ATPase, talk about its mechanism of action, and then go to its functions, and then talk about factors affecting sodium potassium ATPase. That is how you'll get a full mark for your long question if this is coming. All right, so what are the functions of this one? It is electrogenic. What does it mean? So here, what do you see? Sodium is coming inside. And from there, it is going outside. So ultimately, what is happening? Sodium goes outside and potassium comes inside. But more sodium goes outside. So it's as if in net, a negativity is created inside. Because 3 positively charged ions are going outside, 2 positively charged ions are coming instead, creating a net deficiency of 1 positive ion. And that is one of the reasons why we are able to maintain your RMP. Okay, so usually any sort of leaky channels which are there, resulting in any sort of graded potentials that can develop because of influx of sodium or calcium, any positively charged ions. So as long as it does not reach your threshold potential, it comes back down. And that is because of your sodium potassium ATPase. It always comes back down to your RMP because of your sodium potassium ATPase. That is why we call it an electrogenic pump. Okay. Now, another thing is it makes sure that 3 sodium is going out. Okay, sodium is where water follows. So if this was not there, not functioning adequately, what would happen is your cells start swelling up and will eventually burst. So it helps maintain the cell, and as and when required, it can modulate its activity and increase its activity if needed. So ultimately, it helps maintain volume. Now, in your body, sodium potassium ATPase is the major reason why energy is utilized in our body. Okay, so like your basal metabolic rate, for example, is based on your sodium potassium ATPase activity. Okay, and also something else that I was mentioning earlier, sodium potassium ATPase often is important for secondary active transport because this is what creates the gradient for sodium. It takes sodium outside. So that sodium can now come in through a secondary transporter. Understood? So when you talk about the functions, you have to mention it's electrogenic, it maintains volume, it is important for energy utilization, and creates a sodium gradient for secondary active transport. Okay. And then next is factors affecting sodium potassium ATPase. So as I was talking about BMR again, how does thyroid hormone increase your BMR? By activating your sodium potassium ATPase. Okay. So thyroid hormone, aldosterone, insulin. These are examples of increase in the sodium potassium ATPase. So aldosterone in a way increases sodium potassium ATPase in the nephron to result in the activity actually. So anyway, we have your thyroid, we have your aldosterone, insulin, they basically increase your pump activity. On the other hand, substances like dopamine, digitalis can decrease pump activity. This is also an important question. They'll ask you mechanism of action. How does it work? Okay, so where do we use it? So this is used in heart failure. So we have your sodium potassium ATPase. What does it do? It takes sodium, 3 sodium outside and 2 potassium inside. Okay, we have another transporter over here where this sodium can come in and instead calcium will go out. This is the sodium calcium exchange. Okay, this is a sodium calcium exchanger. Now, in heart failure, one of the problems is the heart is not able to pump enough. So how can you increase the contractility? One way we can do that is by increasing the availability of calcium inside. So what we do is we use this to inhibit sodium potassium ATPase. Hence, that gradient is no longer there. Hence, calcium will not go outside. Instead, calcium is going to stay inside, making more calcium available for contractile activity. So that is why digitalis is often used in heart failure. Okay. Now, apart from that, we have hypoxia, hypothermia, and metabolic poisons like 2,4-dinitrophenol, which can also decrease the pump activity. So again, I'm just repeating because this is a very important question. Sodium potassium ATPase if a question is coming. You talk about alpha, beta. Okay. You talk about the diagram, mechanism of action. You talk about the functions and then the factors affecting. Okay. Now, after this, you can talk about secondary active transport. So this secondary active transport, as I was telling you, it can either take substances along the same direction or in the opposite direction. So it can come either as a co-transporter or a counter-transporter. We often call this symporters or antiporter or simply exchangers. We have a co-transporter, counter-transporter. You just need to give some basic examples for this. Okay, as well as your primary active transport. So one example of primary active transport is your sodium, potassium, ATPase. Okay, next is the proton pump, which is there in your acid secretion. Okay, so that is your hydrogen, potassium, ATPase. And then beyond this, we have a circa. which is a calcium ATPase. Okay, so these are examples for active transport. Now, in co-transporter, what is going to happen? Some sort of sodium potassium ATPase activity, sodium goes outside. Now, sodium comes inside through another transporter, but along with sodium, something else comes in. So, a common example for this is your SGLT, sodium glucose co-transporters. Okay, so what happens? Sodium potassium ATPase is there. Sodium goes outside. Now sodium comes inside. Now, along with sodium, something else comes inside, like glucose or amino acid or iodide. Okay, so these are in various places. This glucose is just your SGLT. Apart from that, sodium, amino acid co-transporters are there, sodium iodide co-transporters are there for iodide trapping in your thyroid. Apart from this, we also have counter transporters. One example of counter transporters has already been explained over here. Remember the sodium calcium exchanger. Here, what did happen? Sodium came in one direction, calcium went in another direction. Okay, so this is an example of a sodium calcium exchanger becoming an example of a counter transporter. Okay, so also like this, we also have a sodium hydrogen exchanger also in many parts. Okay, so with examples now, you can explain all of these things. So, first question, enumerate various transport mechanisms. We already mentioned that. Okay, explain active transport with an example that is also done. Now, one thing which I have not mentioned, again, based on the marks also. So, apart from this simple and active transport, we also have vesicular transport, where you'll have to talk about exocytosis and endocytosis. Okay, please note, again, when, a 7-mark question or 5-mark question or 10-mark question, and the question is enumerate and explain the various transport mechanisms, I see that students often talk about simple and passive and active, but forget to write about vesicular transport mechanisms. Okay, in which scenario you'll have to talk about the transcellular, paracellular, and you'll have to talk about how exocytosis, endocytosis, clathrin mediated, caveola mediated mechanisms and all that you have to talk. Phagocytosis. Okay, pinocytosis. So all of these are examples over here. So this is something you should not forget. All right. Now, facility diffusion, again, we mentioned, define, and factors affecting. So factors affecting your diffusion is another often asked question. So that can be easily explained by one simple formula, that is the Fick's law. So Fick's law states that the rate of flow. Okay, the rate of flow is directly proportional. Okay, the rate of flow is directly proportional to concentration gradient. The more the concentration gradient, more the rate of flow. Okay, into area by thickness of the membrane. What does it mean? When the surface area through which it is passing through is increasing, rate of flow increases. On the other hand, if the thickness of the membrane is increasing, rate of flow decreases. So here itself, you have 3 factors. Okay, so we have concentration gradient, we have area, and we have thickness. Now, concentration gradient can also be replaced by pressure gradient. Okay, the nature of the substance, whether it is lipid soluble, it is water soluble, okay, that determines the rate also. Okay, the nature of the membrane also. The electrical. So this is very important when you talk about your Nernst potential and all that. So it's not only the concentration gradient, but your electrical gradient also can further impair this. Okay, so like this, you can talk about your Fick's law and try to explain some of the factors which affect. Next, we can talk about phagocytosis, exocytosis, endocytosis. So phagocytosis, you can talk about in these settings. Detection, internalization, phagosome formation, and phagolysosome formation. Okay, so for phagocytosis take place, generally the size of the object should be more than 0.5 micrometer. So substance more than 0.5 micrometer, and we have a cell over here, and this is what needs to be engulfed. Once it goes close and there is a receptor and binds to it, first step of detection is done. Then pseudopods start coming out and it gets internalized. Okay, when it gets internalized, for phagosome formation to happen, what is going to happen is there'll be minimal water. Okay, fluid won't be there. Instead, it'll be more localized. So this substance that is being taken, it'll be more localized and it goes inside. And a small vesicle like this with the substance will be present with very minimal fluid. Okay, once this is done, now the lysosomes which are here will degranulate into this to form your phagolysosome and destruction of it. And parts of it can later bud off. This is phagocytosis. Okay. One main thing before that is the size of this, the object must be more than 0.5 micrometer. Okay. Now we'll talk about the various methods of endocytosis. Okay. We have phagocytosis. Apart from that, we have pinocytosis and non-phagocytic pathways. So we have phagocytosis. Then we talk about pinocytosis. In pinocytosis, we have micro and macro pinocytosis. So remember the size of more than 0.5. When it's less than 0.5, around 0.2 to 0.5, it is macro pinocytosis. And when it is less than 0.1 micrometer, that is micro pinocytosis. Okay, so ultimately, compared to phagocytosis, there's more fluid involved. So you have a cell here, there is a substance over here. Okay, so often what is going to happen is a head like this comes out and it internalizes it. Now there is a lot of fluid along with this. So this is why it's called cell drinking. Okay, so this is an example of your macropinocytosis. In micropinocytosis, a small particle just comes in along with a endocytes with vesicle with fluid along with it. And usually for this, some mechanisms of, clathrin is involved, caveolin. Okay, so this is an example of how micropinocytosis are. Now we'll come to clathrin and caveolin mediated mechanisms. Okay, so this is again something that students fail to answer well. So there are two mechanisms, clathrin and caveolin. Okay, so can you see over here? There is a receptor. Okay, and a ligand is attaching itself to a receptor, because of which one particular triskelion structure comes and attaches to this receptor. So we have a ligand, a receptor, and a triskelion structure, and they all come and attach to each other. Okay, and once they come and attach, this will pull it and it'll bud off. So this is what we see. This is completely budding like this. And then some GTPase activity will come, because of which this will completely bud off as a separate coated vesicle. So we have a triskelion structure, okay, of clathrons. Clathrons, we have a triskelion structure, which will now come and form a lattice network around, which will help bud the ligand attached to the receptor, and a coated vesicle comes out. Now later, with the help of energy, the vesicle gets uncoated and the triskelion comes out. Okay, so this is an example of how your clathrin-mediated mechanism happen. And ultimately, they go and combine to a particular structure called the endosome, where further lysosome can come and bind. Okay, so this is your clathrin-mediated mechanism of endocytosis. And beyond this, we have caveolin-mediated. In caveolin-mediated, there is usually a lipid-rich area which would be there. Okay, in that lipid-rich area, can you see here? This sort of a structure will be there. A GPI-anchored protein will be there, and there will be a substance called caveolin. Now, again, in a similar fashion, can you see this GPI-linked receptor molecule, and the ligand attaches, and through caveolin, again, a caveolin-coated endocytic vesicles will be. Now, instead of endosome, this goes to a structure called caveosome. That is the essential difference. Okay, otherwise, pretty much they're similar how they act. Okay, this is otherwise in a lipid-rich area. Okay, so here can you see a example of a clathrin? So in clathrin, this clathrin will coat it and come in, and it will attach itself to an endosome and later your lysosome. On the other hand, here caveolin-mediated endocytosis. These are all diagrams you can draw and explain. So when you have to talk about endocytosis, now we have phagocytosis, we have pinocytosis, we have non-phagocytic pathways, including clathrin and caveolin mechanisms. Okay, so in endocytosis, you can mention that. In exocytosis, it is pretty much similar. How you explain your mechanism of neurotransmitters, for example. Okay, so a vesicle will be there. Now it has to dock first. Okay, calcium should come inside. Okay, so there's a B-type calcium channel. Calcium comes inside. It'll bind to a particular substance called synaptogmin, which will result in configurational changes. Okay, we have a T-SNAP protein and a V-SNAP protein. V for vesicle. Okay, so this V-SNAP protein binds to the T-SNAP protein. Docking happens, and another molecule called synaptophysin helps fuse the vesicle to the membrane, and finally an exocytosis can happen. Okay, so that is how exocytosis can happen. And attached to it are various proteins where variable degree of NSF activity and ATP activity and all can happen. So that is how you can mention exocytosis with a diagram. Okay. Now again, factors affecting rate of diffusion across cell membrane, we already talked about it. Fick's law, and you describe the factors. Function of calcium ATPase. So calcium ATP is something we saw. It's also called SERCA, sarcoplasmic endoplasmic reticulum calcium ATPase. What is its main function? The main function of that is, you can see over here. So in your muscles, your dihydropyridine receptors, rhinode receptors, ultimately calcium comes outside. Now that calcium which comes outside should be actively taken back into your sarcoplasmic reticulum. That is by this particular protein, which is SERCA. So what does it do? It actively, there's a lot of calcium inside this. So actively, it transports calciums inside back to your sarcoplasmic endoplasmic reticulum. Okay, so that is a function. You can draw this diagram also and explain this. Name a voltage gated channel. and ligand gated channel. What is a voltage gated channel? So channels, ion channels can be gated or ungated. Okay. So voltage gated means it opens up or closes up based on a particular voltage level. Ligand gated means if a ligand comes and attaches, then it opens up. Okay, a classical example of a voltage gated channel is a voltage gated sodium channel. When the threshold level of your action potential is reached, voltage gated channels are opened up. Voltage gated sodium channels, to be more precise. That will result in action potential. On the other hand, an example of ligand channel, there are many ligand gated channels. One example, for example, if you're talking about your inotropic receptor, okay, you can talk about your nicotinic receptor and N type of nicotinic receptors. So your acetylcholine comes, binds to your nicotinic receptor. That is the ligand, acetylcholine, based on which a channel is going to open up, resulting in sodium influx. Okay, so that is an example of a ligand-gated channel. All right. Now, one small thing that I need to top this off with is your apoptosis, because again, this is put in the new curriculum. So if you look at previous questions, a lot of questions might not be seen in various question papers, but recently they've been asked to know about apoptosis. Okay. So in apoptosis, you will have to talk about so in apoptosis, you have to understand one, it is a gene-driven activity. Okay, it requires ATP. But the cell membrane integrity is not compromised, it is maintained, and there is no inflammation. So here, what is happening is one cell is there are a lot of cells nearby. Okay, it's not like all the cells nearby are affected. There's just one cell in a gene-driven ATP-mediated mechanism that is destroyed. But it's not like its membrane is damaged and it completely goes off and causes inflammation everywhere. That is not how it happens. It is only localized to that particular cell. Okay, the membrane is still intact, and then slowly it comes off at small apoptotic bodies and dies off. So for this to happen, there are two mechanisms, one from outside, one from inside. Okay. Extrinsically, if certain substances come and combine to a particular receptor, like a death receptor, that will activate your caspase 8. So always there's going to be an initiator caspase and an executioner caspase. So this example of caspase 8 is a initiator caspase. So outside, when a fast ligand gated death receptor is activated by a ligand, it will activate a caspase, which is an initiator caspase, which is caspase 8. Okay. And in a similar way, inside the body, inside the cell, okay, mitochondria is one level where you can detect damage through a particular channel called the MPTP, or a nuclear damage, which can go indirectly and tell the mitochondria as well. And what kind of nuclear damage? Say, for example, there are particular gene-driven mechanisms which are there. Okay, so we have a factor, transcription factors like BAX and BCL. This BAX is pro for your apoptosis, and anti-apoptotic is BCL. Okay, so based on how much of it is there, how much of its expression is there, that will help us understand about your nuclear damage. So either nuclear damage or mitochondrial damage indirectly will activate another caspase, which is an initiator caspase. An example for that is caspase 9. So why either a caspase 8 through an extrinsic factor or some intrinsic mechanism, caspase 9 will ultimately activate a common pathway where caspase 3 and 7 is activated. That is your executioner caspase. And when that comes, that is when your apoptosis is activated. Okay. Now, if you have to talk about the functions of apoptosis. So again, apoptosis, what is apoptosis? Why is it different from other mechanisms? What are the mechanisms involved here? And what is the function? Again, function is very often asked. They'll just directly ask you what is the function. So one, in embryogenesis. In embryogenesis, what happens? Especially in your limb digits formation, a mold comes, and from there, through apoptosis, your digits, by removing parts in between, your digits are formed. Now, again, between a male and a female structure. So when we are born, we're both born with both male and female structures. Okay, when you become a male, the Mullerian structures needs to regress. That is again through your apoptosis. Now, immune selection, clonal selection. Okay, if, for example, there are particular cells, okay, WBCs, which react to self-antigen, then how do you destroy it when it reaches thymus, for example? That is through your apoptosis. Menstrual cycle, when shedding happens, that is also through apoptosis. And again, intracellular organisms like virus or some sort of prion or like, other misfolded proteins. Okay, you need to destroy such dangerous cells without affecting everything around it. That is again through apoptosis. Okay, so these were the various questions that we had to cover over here. Okay, very important topics. Entire general physiology, this is the most important topics actually. I'd want you to know it thoroughly for your exam. Okay, so just to brief you about the features of Auricle, we got live sessions, recorded sessions, and there are notes which are readily made available for you, and MCQs are also present. And these are various subscription plans that are available. You can use my code for discount on subscription. Okay, and you can ask doubts, or if you want to connect with somebody, you can connect with the social media handle for Auricle. Okay, so thank you.